Okay, in the second part of chapter 4 on the cell, we're going to be comparing typical eukaryotic animal and plant cells. So this is just going to be a generalized survey looking at some of the similarities and the differences between the two. There's figures in your textbook, there's figures in your lab manual, as well as these PowerPoints and that are loaded in Brightspace for you to practice learning these portions of the cell and their respective functions. So let's just look at a typical animal cell first. This is the image from your textbook. We can see the animal cell's outer boundary forms this plasma membrane. And in many instances, the plasma membrane in areas where surface area needs to be increased even more will have these undulations or folds called microvilli. Now, all of the material inside of the plasma membrane is referred to as the cytoplasm or the cytosol, and floating or suspended in it amongst the cytoskeleton are the various organelles and the nucleus. So if we look at the parts of the nucleus, we see that the nucleus's outer boundary is called the nuclear envelope. It's a double folded membrane, and there are protein-lined perforations in this nuclear envelope called nuclear pores, which when opened and closed will allow materials to enter and exit the nucleus. The genetic material on the inside of the nucleus is usually in the form of chromatin when the cell is functioning normally, and chromosomes when the cell is getting ready to divide. So this is the genetic material. And then finally, there's a structure called the nucleolus. And the nucleolus is a darkened area in the center of the nucleus. Some cells have more than one nucleoli in their nucleus. And the nucleolus is the site of many parts of the ribosome formation. Now, if we look out in the cytoplasm, we'll see these ribosomes. And these ribosomes are these very small dots found either as free ribosomes in the cytoplasm or attached to the rough endoplasmic reticulum. So the endoplasmic reticulum is this tubular network of membrane that extends through the cytoplasm, and with the ribosomes on it, it forms the rough endoplasmic reticulum, and this is going to be a location where a lot of protein synthesis is going to occur. Of course, the job of the ribosomes is to help incorporate or help translate that genetic code from nucleic acid language to the language of proteins, which are the sequence of amino acids. Now, we also see smooth endoplasmic reticulum. The smooth endoplasmic reticulum is again this tubular network of membrane, but it does not have ribosomes on its surface. And the smooth endoplasmic reticulum's function is to create detoxifying agents or lipids. Now, a packaging unit is this pancake-like Golgi apparatus or Golgi complex, and we'll learn a little bit more about this when we start talking about transporting the cell. But this is where a lot of the membrane proteins or other proteins are processed here. So as they go through their tertiary and quaternary structure, the transport vesicles originate from the Golgi complex, and these vesicles are sometimes allowed to merge with the outer plasma membrane during this process called exocytosis, and this is where things are going to be released from the cell. Now, the energy in our animal cell is derived by ATPs, and ATPs are converted in large numbers in this organelle called a mitochondrion. The mitochondrion has two basic parts to it: this inner highly folded membrane called the cristae, which surrounds this fluid and enzyme filled matrix. And when we talk about aerobic cellular respiration in Unit 3, we'll learn all about how the mitochondria is put together to accomplish this job. The Cytoskeleton is the network of proteins in an animal cell that helps to give the animal cell its shape. And so you'll find a lot of these cytoskeletal proteins in the cytoplasm, some attached to the membrane and some just in here for support. Another cytoskeletal element is this structure right here called a centrosome. And a centrosome consists of a pair of centrioles. These centrioles are going to split during the process of cell division, and they'll help provide the mitotic spindle that will be used as the cell gets ready to divide. Now, some other structures that we'll find in these cells are these larger storage things. These are vacuoles, and vacuoles can have lots of different functions. For instance, this large vacuole is filled with digestive enzymes, and it plays a crucial role in digesting materials inside of the cell or in actually recycling cellular components. Because it's filled with digestive enzymes, this is called a lysosome. While this particular round vacuole is filled with this peroxisome is filled with agents that help to break down peroxides. Enzymes called catalase, which are important to help detoxify the hydrogen peroxide that's produced inside of cells. You might also see some food vacuoles or storage vacuoles inside of the cell as well. So this is the basic parts of our animal cell.